Alright everyone, welcome back to our next lecture presentation. In this video we will cover the digestive system. We're going to get started with the anatomy of the digestive system, and then we'll work our way into some of the physiology of the accessory glands and organs that can be found in the digestive system also. So as it sounds, the digestive system is primarily responsible for digesting food, but there's quite a lot that actually goes into digestion as a process. Digestion can be broken down into both mechanical digestion and chemical digestion, both of which I will explain very shortly. But in order for us to adequately grasp what the digestive system does, we must first discuss the anatomy of the digestive system and get an idea of what components make this system up. So structurally, the digestive system is a long tube that leads from the mouth to the anus. There are lots of components that contribute to the tube known as the alimentary canal. That is the tube from the mouth to the anus. So components such as the esophagus, stomach, small intestine, and large intestine are all parts of the alimentary canal. They make up part of that tube. But the digestive system in particular contains lots and lots of accessory organs that will aid in the process of digestion and absorption also. Just to name a few, the liver, the pancreas, and the gallbladder are accessory organs. Those organs will never contain food. Food will never go into those organs to get broken down or digested. However, those organs play an integral role in the entire digestive system as they produce digestive enzymes that will aid in the actual digestion that takes place inside of the alimentary canal. Other accessory structures, such as the teeth and the salivary glands, will help to break food down into a paste that is much more manageable inside of the digestive system. This is the process known as mechanical digestion, the breakdown of large pieces of food into smaller pieces of food. Now, it's those accessory organs that are releasing digestive enzymes that are going to facilitate the process of chemical digestion. Chemical digestion can be described as the actual chemical breakdown of macromolecules into their smaller, more simple parts. We also have the process of absorption that is also going to play a big role with the digestive system. Digestion would be totally pointless if we couldn't absorb the smaller digested nutrients into our bloodstream so that we can process them and eventually use them for energy. So it is digestion and absorption that are the two major functions of the digestive system. We're going to elaborate much more on this throughout the course of this presentation. We're going to talk about the structural components of the digestive system and what function each of them carries out. And we're also going to discuss some digestive physiology and see which enzymes are released from where and what those enzymes are going to break down. So with the digestive system in particular, there's often lots of words that get confused. So we want to make sure that we get our definitions straight before we start jumping into the technical terms that we're going to be covering in this lecture presentation. So first and foremost, we have a couple words on this slide: ingestion, deglutition, and peristalsis. You may have heard of one or more of these terms before, but we are going to specifically describe and define what each of these words mean. Ingestion is the actual process of putting food into the digestive system. It is during this process that we start mechanical digestion and also chemical digestion as well. Since mechanical digestion refers to the actual physical breakdown of food, we typically would consider mechanical digestion to be the major process that takes place when food enters the mouth, as we chew our food, mix it with saliva, and manipulate it into a form that we can easily bring into the digestive system. However, one thing that we are going to see when we get into digestive physiology is that there are enzymes in the saliva that will already start the process of breakdown as soon as food enters into the mouth. So chemical digestion will take place as food enters the mouth also. Deglutition is a fancy word for swallowing. This is the process of moving food from the mouth into the esophagus. We do have several phases of swallowing, and not all of them are voluntary. Some of them are carried out by the esophagus. But deglutition refers to the action of moving food down into the esophagus. Once food has passed into the esophagus and our voluntary muscles are no longer influencing the bolus that has entered into the digestive system, it's time for our smooth muscles to take over and propel those contents along the alimentary canal. And that's where peristalsis comes in. There is a layer of smooth muscle that can be found in the walls of the organs that make up the alimentary canal. And this smooth muscle will contract and move food along the digestive system. There are in fact two layers of muscles that will sit right next to each other, and we will discuss those specifically when we get to the layers of the alimentary canal. This layer in particular that contains these two sublayers of muscles is known as the muscularis externa. So as we bring food into the digestive system, we call that ingestion. As we swallow food, we call that deglutition. And then the continuous movement of substances through the rest of the digestive system we call peristalsis. So to ensure that you are on board with the structural organization of our digestive system, we're going to go over the components of this system on this slide. So again, keep in mind that the alimentary canal is the long tube that goes from the mouth to the anus. And there are many different components that make up different parts of the alimentary canal. We can go ahead and start with the mouth. The mouth, of course, contains the oral cavity. But inside of the oral cavity, we have the tongue and the salivary glands. We have three salivary glands: the parotid gland, the sublingual gland, and the submandibular gland. The tongue, of course, is also found in the mouth, and that will aid in the process of mechanical digestion. The tongue is very useful in manipulating food, and along with the saliva from the salivary glands, we can turn solid food into a paste-like substance. This is what we call a bolus, and that's much more manageable when we want to swallow food and bring it into the esophagus. So when you do swallow food, that's going to enter into the esophagus. The esophagus is found just behind the trachea, which we saw during the respiratory presentation. And the esophagus is going to have some layers of smooth muscles in its walls that are going to help bring food down into the stomach. It takes about 1 minute for food to enter into the esophagus and make its way down into the stomach. And the stomach can be found here. The stomach is a storage site for food, so all of the food that we swallow will end up in the stomach, and it will slowly be released into the small intestine. The small intestine is the lighter tube that can be found over here. And specifically, the stomach will dump a substance known as chyme into the small intestine. As we are mechanically digesting food, we call it a bolus. But inside of the stomach, there's hydrochloric acid, meaning that the pH is extremely low and very, very acidic inside of the stomach. This very low pH environment will aid the process of chemical digestion, and the bolus that has entered into the stomach will be further broken down into a substance known as chyme. So the stomach will allow this chyme to enter into the small intestine, of which there are three parts. Specifically, the three parts of the small intestine are listed here. The first part, which is where the stomach dumps chyme into, is known as the duodenum. The duodenum comes straight from the stomach and eventually will dump into the second part, known as the jejunum, and the jejunum will direct food into the third part, known as the ilium. And so the ilium can be found here at the terminal part of the small intestine, and it dumps into the large intestine, which can be found coursing here. The large intestine will have some of its own parts, and we will see those on the next slide. Here at the beginning of the large intestine, we're going to find the vermiform appendix that sticks off here, and the very end of the large intestine is referred to as the rectum. So this characterizes all of the structures that can be found in the alimentary canal. From the mouth to the anus, there's quite a few different places that substances are going to have to go to be digested and eventually be absorbed. Now, I've just described the components of the alimentary canal, but there's some other things on this slide that I want to make sure that we mention as accessory structures. So here you can see this organ is the liver. The liver is going to be very, very important for digestion because it produces a substance known as bile. The liver will release bile, and it will eventually get stored in the gallbladder. The gallbladder's main function is to store bile. In fact, the gallbladder does not make bile at all. It only stores bile that comes from the liver. Bile is going to be very useful in helping to break down fats. Now also back here, not labeled on the slide, is this little orange and spongy looking organ that's found here behind the stomach. This organ is known as the pancreas, and the pancreas is one of my absolute favorite organs that we have. The pancreas is one of my favorites because it has both an endocrine function and an exocrine function. But here in the digestive system, we need to focus on the exocrine portion of the pancreas. So we will come to see a bit more about the actions of the accessory organs, but let's make our way back to the large intestine and discuss the individual parts that can be found on this organ. So you can see that this slide has zoomed in on the large intestine, and there's quite a bit that we're going to be able to see from this slide. But first and foremost, I want to make sure that we understand what this structure is here, known as a mesentery. Mesenteries are part of the serous membrane of the digestive system, known as the peritoneum. I will have some pictures of the mesentery for you in just a few slides. But this structure is the serous membrane that our large intestine is suspended within. Inside of the mesenteries, we'll find lots and lots of blood vessels as well as lymphatic vessels that will carry dietary fats away from the digestive system. You can see the different parts of the large intestine now labeled on this slide. So the small intestine comes directly from the stomach. We have the duodenum, the jejunum, and the ilium, respectively, making up the small intestine. The ilium, being the last part of the small intestine, will eventually take chyme over here to the large intestine. And so over in this area, we're going to have what's known as the ileocecal valve. The ilium is going to come over here and dump chyme into the first part of the large intestine, which is known as the cecum. That is this portion here. This first portion of the large intestine is the cecum. Now, based on the direction of substances inside of the large intestine, we name the different parts. So you can see that once substances enter into the cecum of the large intestine, they're going to get forced upwards, and therefore we call this the ascending colon. As we make our way up, we're eventually going to cross over the body, and this is where we have the transverse colon, as food will move across the body. And then this portion of the large intestine is the descending colon, as that's going to take food downwards into the sigmoid colon. And the sigmoid colon is the last part of the large intestine that's roughly shaped like an S. This will take stool into the rectum, and you can see the rectum is the very last part of the large intestine. From here, we've made it to the end of the alimentary canal, and therefore the external anal sphincter that's found here will be controlling the elimination of wastes from the digestive system. So the large intestine marks the last component of the alimentary canal. While all of the components of the alimentary canal are structured very differently, they will have some things in common. And one of those is their layers. There are 4 layers that can be found in the alimentary canal, each layer containing different things. Specifically on this slide, we're looking at what seems to be the small intestine. However, these layers can be found in the stomach, the esophagus, the large intestine, and the small intestine as well. They will look a little different depending on where we're at in the alimentary canal, but all of these layers are preserved throughout each of the structures that make up the actual alimentary canal itself. So by definition, the first thing that any organ needs in order to be considered a part of the alimentary canal is the lumen. Again, since the alimentary canal is the long tube from the mouth to the anus, that means that there has to be some passageway for substances to make their way along that pathway. So the lumen is the first thing that we will see as the space inside of the digestive system that will contain food. Now, the layer that's found adjacent to the lumen is known as the mucosa, and the mucosa in particular contains 3 other layers inside of it. As this is a cavity in our body, we will have a layer of epithelial tissue that is unique to the digestive system. We will also find just deep to the epithelium the layer of connective tissue under here that's known as the lamina propria. The lamina propria is a connective tissue layer made up of areolar connective tissue, and there's many blood and lymphatic vessels that can be found in this connective tissue layer. It is from these blood vessels and lymphatic vessels in the lamina propria that nutrients will be absorbed from the digestive system. We also have a layer of muscles outside here in the mucosa known as the muscularis mucosa. This muscular layer creates different folds to increase the surface area inside of the digestive system for absorptive purposes. Now, wrapping around the mucosa, we have what's known as the submucosa. And the submucosa is entirely made up of connective tissue. This is also areolar connective tissue containing many blood and lymphatic vessels that will aid in carrying nutrients as well as the vessels that can be found here in the lamina propria. Now, as we continue to move further out, we will find the muscularis externa. This is a very special layer that contains 2 sublayers of muscles, one circularly arranged and one longitudinally arranged. You can see from this slide that the inner sheet of muscle is circularly arranged and the outer sheet of muscle is longitudinally arranged. As we saw with blood vessels, circularly arranged muscles will aid in decreasing the diameter of the lumen. Therefore, when this inner circular layer of muscles contracts, that's going to segment substances into different pieces. However, here in the digestive system, we're also going to have a longitudinally arranged layer of muscles also. The longitudinal arrangement will aid in shortening the entire length of the digestive system, therefore helping to propel substances along the alimentary canal. It is this outer longitudinal layer of muscles that will facilitate the process of peristalsis. On the outside of the muscularis externa, we'll have the serosa. As the name implies, serosa means serous membrane. Therefore, we also call this layer the visceral peritoneum. There is also areolar connective tissue here, but also a layer of simple squamous epithelium, as a serous membrane is considered a simple organ. You can see right here, you've got the simple squamous epithelium and the areolar connective tissue that can be found just below it. Now, this serosa is going to wrap around the entirety of the alimentary canal. But we must remember that the organs of digestion are suspended within the peritoneum. And so you can see that we have these two layers of the serosa that are coming back to meet. As this part of the alimentary canal is suspended in that peritoneum, we're eventually going to have those two parts coming back together. And it's right here that we're going to give this the name, the mesentery. You can see the blood vessels and the lymphatic vessels that are coursing out to the digestive system through the mesenteries. So while there are many different layers, and while these layers will look different in different parts of the digestive system, most of the components of the alimentary canal will be structured with these layers and be arranged with these layers in mind. We mustn't forget our accessory structures, and that's what we'll see on this slide. While the gallbladder is not part of the alimentary canal, it will aid in digestion. The liver is going to produce bile, and the gallbladder will store it. That's very important to understand. The gallbladder does not make its own bile, it only stores bile, so that we can have it ready for when food makes its way into the small intestine. Here you can see the duodenum, the first part of the small intestine, and the jejunum that's going to connect the duodenum to the ileum. Oftentimes we describe the small intestine as hugging the pancreas, and you can see that arrangement right here. The pancreas is going to have this long duct that can be found inside of it, and right here we're going to be releasing digestive enzymes from the pancreas and bile from the gallbladder through what's known as the hepatopancreatic ampulla. As food moves its way out of the stomach, it's going to end up here in the duodenum, and this is one of the major locations that digestion is going to take place in our body. So on this slide, we're going to talk a little bit more about the peritoneum. As was discussed in Anatomy and Physiology 1, there are three serous membranes that we have inside of our thoracic and abdominal pelvic cavities. Two of them are found up in the thoracic cavity, which will refer to the pericardium that wraps around the heart, and the pleura that wrap around each lung. Both of those are found in the thoracic cavity. But in the abdominal pelvic cavity, we will find the peritoneum. The peritoneum is the largest serous membrane in our body, and oftentimes the peritoneum looks rather intimidating because it wraps around so many different things. But we must remember that the peritoneum is still a serous membrane, just like the pleura and the pericardium. It's going to wrap around organs and form a parietal and a visceral layer. You can see the peritoneum being shown down here. This specifically is the parietal layer of the peritoneum, or the parietal peritoneum for short. As the peritoneum starts to fold back in on itself, it's going to contain lots and lots of parts of the small intestine. That's what we see here. These small intestines are going to be suspended within this. We'll see the large intestine suspended within this, and also the stomach. Now, where the peritoneum starts to fold in on itself is where we consider it the visceral peritoneum. So this here is the visceral peritoneum that's wrapping around our intestines. And to be even more specific, where these two layers of the peritoneum have come back to touch each other, that's where we have our mesentery. It is in the mesentery that we'll find those blood vessels and lymphatic vessels coursing out to supply the organs that are suspended within the peritoneum. In particular, there's one very large fold that can be found on the peritoneum, known as the greater omentum. The greater omentum is the longest peritoneal fold, and it's going to cover part of the large intestine and the small intestine, sitting more anteriorly to the rest of the digestive structures. Oftentimes, lots of adipose tissue will be found attached to the greater omentum and give us the characteristic beer belly that is seen in some overweight individuals. You can see here that the omentum has been pulled upwards. We need to move this out of the way so that we can see the large and small intestine. And you can see the greater omentum also shown right here, and the adipose tissue that is associated with it. From this picture, you can see the liver right here. You can see the gallbladder that sits just under it. Remember, the gallbladder does not make bile, it just stores bile. Here is going to be where the esophagus comes down and takes food into the stomach. And the stomach will then flow into the small intestine, which we can see here. We must remember that the small intestine will be covered up with the greater omentum when this is draping downward, as it normally is. We'll find the spleen that's tucked up here in the left hypochondriac region. This is going to be found just under the diaphragm. And the first part of the large intestine, known as the cecum, that can be found over here. This will make its way up, and it will course across the body. Again, this is covered up by the greater omentum that's positioned right here anteriorly. Now, to get a better view of some of these structures, we're going to start looking at these structures on the cat. This picture will show us what the body looks like when the greater omentum is draping over the small and large intestine. You can see the greater omentum here and all of the adipose tissue that can be found on it. Once again, the greater omentum is the longest peritoneal fold. We'll also find the liver that's tucked up here under the diaphragm. This is releasing bile into the hepatopancreatic ampulla and also allowing the gallbladder to store any excess. Just under the liver, we will see the stomach, which is receiving food that is swallowed from the esophagus. You will also see the spleen here that's found on the side. This is primarily going to contribute to our immune system. Once we take the greater omentum and move it upwards, that will expose the small and large intestines. The small intestine is actually much longer than the large intestine is, but we call the large intestine because its diameter is much bigger. The second part of the small intestine is the jejunum. The jejunum in particular is a very absorptive part of the small intestine. We see the duodenum coming down here from the stomach and hugging the pancreas. The pancreas often looks rather glossy and has a silverish appearance. The duodenum will eventually take chyme into the jejunum. We've seen this picture once before. You can see the small intestine shown here with the mesenteries taking blood vessels out to the small intestine. These blood vessels are extremely important for supplying oxygen to the muscles in the small intestine. Remember, the serosa is also known as the visceral peritoneum, and out here we'll find the parietal peritoneum. This parietal peritoneum is eventually going to fold back on itself and form the layers that make up the mesentery. The last part of the small intestine is the ileum. The ileum will project out from the jejunum and eventually take substances into the large intestine. Here we will find the large intestine, specifically the cecum of the large intestine. This is the part of the large intestine that receives substances from the ileum, and the cecum will then project out the ascending colon. Based on the direction of movement of substances inside of the large intestine, we will have the ascending colon here, and then the transverse colon will course across the body. Just so that we're aware, the large intestine can also be called the colon. When we move the abdominal contents to the right side, you see that we'll expose the descending colon. The transverse colon will take substances across and eventually enter the descending colon as substances will move downward back to the sigmoid colon, which will eventually take stool out of the anus. So now that we've discussed the structure of the digestive system, the components that make up the alimentary canal, and the accessory organs that contribute to the digestive processes of the digestive system, we're now going to transition into digestive physiology and talk a little bit about how digestion takes place. So as discussed at the beginning of this presentation, digestion can be separated into mechanical digestion and chemical digestion. Mechanical digestion is the actual physical breakdown of substances. The teeth are major components that facilitate mechanical digestion. They take large substances and turn them into smaller substances. Eventually, as we mix food with saliva, we form this paste, which we call a bolus. So truly, we can describe mechanical digestion as the conversion of food into a bolus. Now, chemical digestion is a bit more intricate. The entire purpose of digestion is to break food down into small enough pieces that we'll be able to absorb it, and eventually use it. In order for us to absorb substances into the body, they must be small enough in order for us to do this. So chemical digestion is what's really going to facilitate the more specific breakdown of substances into their monomeric forms. Chemical digestion is primarily what we're going to discuss here with digestive physiology, and we'll see that enzymes are going to play an extremely important role in doing this. Just as a reminder, enzymes are going to bind to a substrate and catalyze a chemical reaction. So it is during chemical digestion that enzymes are going to take the macromolecular forms of substances and break them down into simpler substituents. One other characteristic that we need to understand about the alimentary canal are the intestinal folds. There are several degrees of folding of the intestinal system. You can see that grossly, the intestinal walls will fold in on itself to increase surface area. Over here, we'll have these long finger-like projections known as villi that will project into the lumen of the small intestine. The villi are lined by the epithelium of the intestinal walls, and you can see that on the villi we'll have these finger-like projections known as microvilli. And so with all of these extra degrees of folding, we've significantly increased the surface area. And the whole purpose of this is to improve the efficiency of absorption. Microvilli are often referred to as the brush border, and there are enzymes that can be found on the microvilli that will play an extremely important role in digestion. So we're going to transition here specifically into mechanical digestion, and then we're going to discuss the different processes that compose what we know as chemical digestion. Mechanical digestion, as I've explained before, is the physical breakdown of food into a bolus. The teeth play a lead role in mechanical digestion. However, mechanical digestion doesn't truly stop with the teeth. As the bolus is swallowed and brought into the digestive system, the muscles of the stomach, small intestine, and the large intestine are going to further grind up food and mix it in with the digestive enzymes. We must remember that outer longitudinal layer of smooth muscle will facilitate peristalsis. This is the actual movement of substances along the alimentary canal. But we also have that inner circular layer of smooth muscle. This will facilitate segmentation. When different parts of the alimentary canal decrease their diameter, that's going to separate substances into pieces. So it's not just the teeth that facilitate mechanical digestion. The smooth muscles of the alimentary canal will also help to churn up food and mix the digestive enzymes into the contents of the digestive system, and that will also help to accelerate the process of chemical digestion. Now specifically, we're going to discuss chemical digestion in terms of the different macromolecules. Let's take a quick second to review the 4 macromolecules that our body's going to have. We have carbohydrates, proteins, lipids, and nucleic acids. Carbohydrates are often referred to as polysaccharides, and they're made of individual simple carbohydrates known as monosaccharides. Oftentimes, when we eat carbohydrates and bring them into the digestive system, they're in the polysaccharide form, and so we'll have specific enzymes that will break them down into their monomeric structures, which is the monosaccharide. We must also remember that proteins are referred to as polypeptides. There's lots and lots of amino acids that are linked in long chains and then folded upon each other in different degrees. When proteins are digested, we're going to break them down back into their individual amino acids. It's very important that we can break these substances down into their monomeric forms, as we can only absorb them in their monomeric forms. Our bodies are unable to absorb large polysaccharides and large protein. We must break them down into simpler forms if we're going to get them into our bodies. The polymeric structure of lipids included a glycerol backbone and fatty acid chains. So we're left with a monoglyceride and 2 individual fatty acids that can eventually either be broken down or reused depending on what our body's needs are. Nucleic acid was the 4th group of macromolecules that our body uses. However, we do not use nucleic acids for energy, so we're not going to discuss their breakdown much further. So in order for chemical digestion to take place, we must have individual specific enzymes that can act on each of these macromolecules. Every single one of these macronutrients will be broken down by their own individual enzymes. We must remember a couple of the characteristics about enzymes that we've learned through anatomy and physiology thus far. Enzymes themselves are proteins that act on a particular substrate and speed up a chemical reaction. We call these catalysts because enzymes can speed up some reactions up to 100,000 times. Proteins, carbohydrates, and lipids will all be broken down by different enzymes, and also in different places. There are certain enzymes that break down carbohydrates that will not be able to break down proteins, and these individual enzymes will be found in different places depending on what their role is. Since protein function is entirely dependent on protein structure, and enzymes are proteins in fact, we must make sure that the structure of our enzyme remains intact. This means that some enzymes will lose their function at certain places in the alimentary canal. Because our enzymes are proteins, they are subject to denaturation, or the unfolding of their molecular structure. This will cause them to lose their function. So depending on the enzyme, they'll have specific parameters of which they'll function within. Therefore, certain enzymes might function really well in a low pH environment, such as in the stomach. But other enzymes will not be very functional in an environment where the pH is so low. Heat is another factor that can denature protein. If the temperature gets too high, that can disturb the chemical bonds of a protein and cause it to unfold. So we must make sure that the enzymes of our digestive system are existing within conditions that will facilitate their proper function. So we will first get started with carbohydrates. As I mentioned, each macromolecule is going to have their own enzyme that breaks them down. And for carbohydrates, that is amylase. If you ever hear me say amylase, that's the enzyme that breaks down carbohydrates. Since we typically ingest carbohydrates in their polymeric forms, amylase is going to be used to break down carbohydrates into smaller pieces. Since carbohydrates are typically ingested in their polymeric form, amylase will be used to break down polymers of carbohydrates into smaller amounts known as oligosaccharides. Oligo means few, so therefore these are still going to be polysaccharide chains, but much smaller carbohydrate chains. Eventually, amylase will be used to break oligosaccharides down into disaccharides. Amylase in particular is found in the saliva and the juice of the pancreas. In particular, we'll have salivary amylase and pancreatic amylase. Once carbohydrates have been broken down into their disaccharide form, enzymes on the brush border are going to continue their conversion into monosaccharides. And it is in this monosaccharide form that we'll be able to absorb carbohydrates. So one experiment that can be done to demonstrate the action of amylase is our iodine experiment. In ANP1, we used starch and iodine to demonstrate the concepts of cell transport. You may remember that starch and iodine, when mixed together, form a very dark violet color. So one way that we can test the action of amylase is by mixing starch with amylase in the saliva. By having two different starch solutions, you can add a little saliva to one of those solutions, mix it up thoroughly, and leave the other one alone. From here, adding iodine to each well will produce different colors. Starch and iodine together form a very dark violet color. However, once we've added salivary amylase into our other well, saliva has started to break down the starch into its smaller forms. Maltose and iodine do not produce that dark blue color. So this is one way that we can test the effects of amylase. So by knowing that there is amylase in the saliva, that tells us that carbohydrates begin their digestive processes as soon as they come in contact with saliva in the mouth. Now moving on to our next macromolecule, we have a very specialized enzyme that's going to break down proteins, and its name is pepsin. Pepsin is an enzyme that's released by the stomach, and it can be used to break down larger proteins into individual polypeptide chains. The pancreas will also play a very big role with digesting proteins as well as amylase. Peptidases are enzymes that come from the pancreas, and we must remember that ASE, that suffix, means enzyme. So our peptidases will be responsible for breaking down the polypeptides that are formed by the breakdown of proteins. Peptidases will convert polypeptides into peptides, and then our brush border enzymes on the microvilli will take those peptides and convert them into their individual amino acids. It is the amino acid form that our body is able to absorb. So one way that we can test the action of the enzymes that break down protein is by mixing meat tenderizer with gelatin. Specifically, gelatin is a protein, and meat tenderizer contains a peptidase known as papain. When we take gelatin and add meat tenderizer to it, it starts to liquefy. This is a demonstration of what takes place in the digestive system when we release pepsin from the stomach and peptidases from the pancreas. There is in fact one other test that we can administer to show the effects of protein digestion. And this test uses a molecule known as BAPNA. BAPNA, or BAPNA, is a dye that contains no color. Normally this dye is clear. However, when it acts as a substrate for pepsin from the stomach, it's going to start producing a yellowish color. So BAPNA is a very large molecule. But pepsin recognizes BAPNA, and it starts to break this molecule into two individual products. One of those breakdown products is a yellowish pigment. So the more pepsin that we mix with the BAPNA, the more intense the yellowish color will become, as pepsin is able to break down more BAPNA and produce more of that yellowish product. The last macromolecule that we're going to discuss is lipase. Lipase in particular will be coming from the pancreas and help to break down lipids. Lipids, as we know, have a polymeric structure of one glycerol backbone with two fatty acid tails. So lipase is going to catalyze the lysis of these two fatty acid tails from the monoglyceride. We will then absorb the fatty acids into the lymphatic system and eventually use them to store energy for later. So that will conclude our presentation on the digestive system. If you have any questions at all, please send me a message. I will see you all in our next lab presentation when we will cover the urinary system.